So, I'm Wenying Shou from UCL. I'm very nervous now because unlike the normal talks I would give where I know my materials really well, for this one, for the majority of the talk, I will cover topics that I'm not expert on. So that reminded me, how do I advance? Sorry. How do I advance? How do I advance?
Slides, maybe.
Yeah.
Let's just stick that. Oh, yes. So 1/4. So I figured out the way. So 1/4, only 1/4 of the talk will be about what I learned from my laboratory. And the second part is sort of like a random walk, right, across this vast landscape of experimental techniques. And I'm certain that many of you know better about the second part than I do. So I want to first start with measurement challenges in a model organism, the budding yeast. And then, so I'll start with this. And now we'll go to the more challenging aspects of dealing with the non-model organisms. So in this system, pointer? Is it laser pointer? Is it laser pointer or is it a pointer? I can use. Oh, it's like this. This is a pointer. Oh, it doesn't matter. So that's fine. I can just. So basically, so we have set up engineered yeast community, in which the two species will engage, the two strains will engage in metabolite exchange. So for L minus A plus, cell L is lysine. So for the mathematicians audience, lysine is essential metabolite. So if you don't have lysine, you don't grow, right? So these cells cannot make lysine. But then there's a mutation that forces them to overproduce another essential metabolite called adenine. And then the complementary strain cannot make adenine, but overproduce lysine. So then the two will cross-feed and support each other's growth. And we call this cosmo for cooperation that is synthetic and mutually obligatory. So because it's yeast, right? So we can make them fluoresce in, say, red and green, right? And we can track population dynamics. So this is population size in logarithmic scale against time, right? And we can see that the red, the green, and the total in black, after initial lag, all grow exponentially. And so we want to know, say, the slope, right? The rate at which individual strains and the community as a whole will grow. And it turns out after some very simple calculation, know that it is a function of the release rates and the consumption amount. So this RA is the femtomole adenine released per cell per hour. And the CA is the femtomole adenine consumed per cell birth, right? So if you do the unit calculation, metabolites released per cell divided by that, it's per hour, and then the same for lysine, square root, so you get per hour, the right unit. So then so the little test I set up for myself is that if I measure these parameters, right, the two release rates and the two consumption amounts, would I be able to predict this slope? So I tried it, and it just won't work. And eventually, we figured out what was wrong. So the first challenge we encountered is that microbial phenotypes can vary with growth rate. So to illustrate how we know this, right, in which we'll pose a challenge for quantitative modeling. And the way we figured this out is we constructed chemostats. So chemostats, you can think of that as like a feeding tube for microbes, right? So basically, if you want them to grow slowly, you drip in slowly. 1, 2, 3. But if you want to grow fast, you drip faster, 1, 2, 3, 4, right? So then they will grow fast. So you basically can control them. You can force them to grow at the rate up to their maximum growth rate. So here's the graph, right? So we're going to measure lysine consumption per birth for lysine requiring cells. And it's the net growth rate. We can tune it up. to at different rates so we can control growth rates. And then what we found is that, right, so if across chemostats at different rates versus the maximum growth rate versus saturated cultures, we get very different numbers, different by, like order, twofold. And then the same goes on for release rate, right? So if we look at the lysine release rate per cell per hour, and this is again across different growth rates, we see it's non-monotonic behavior. So it goes up and crashes down, right? And then when they grow at maximum growth rates, there's no release. So this is orders of magnitude difference in the phenotype, depending on the growth rate. And a second challenge that we find is its rapid evolution can occur during the very measurements that we make. So we came across this from this, right? So we subject the lysine requiring cells in these chemostats that require lysine. So basically we drip in lysine really slowly, right? So they're forced to grow really slowly. And we look at the lysine concentration in the chemostat, right? So the expectation is that, sorry, I don't have time to go through the math, but the expectation that is that you will, the lysine concentration in the chemostat should reach a steady state level, right? It's expected. But what we saw is not this, right? It's just continuous declining of this, suggesting that evolved cells have evolved to depress the lysine concentration in the chemostat. And we tested, indeed, so we take a sample from ancestor, plate them on a plate, and that plate has very low lysine concentrations. And we look at them on the microscope, right? So this is the unbudded cell. This is a yeast cell that has just budded, right? So like looking, basically it's so low that ancestor cells cannot grow. But if we take a sample from chemostat, 40 hours, right? So what do we see? On the same plate, you will see colonies, like a really robust growth. So suggesting these, these a fraction of them have evolved. And so it's a really rapid rising of evolved cells. So if you want to measure the phenotype of ancestors, you have to do your experiments really fast. And then the third lesson is that imaging spatial patterns, whoops, happened, sorry. Sorry. I don't understand why. Okay, the imaging spatial patterns in communities can be challenging. So remember, like we have this cross-feeding, the cooperating pair of strains. We can also engineer, a third strain labeled with different fluorescent, like blue fluorescent protein. And these cells cannot make lysine. So they take up lysine over the green cells, but they don't actually contribute any. So they're regarded as cheaters. So then when we can plate these, we can take a petri dish with no adenine or lysine and plate 3 types of cells randomly and uniformly on the plate. We can very easily get the top, right? So the XY, if you view from the top, you can immediately see the cooperators are mixed, but the cheaters are segregated. But then still it's not satisfying, right? Because if we actually want to know what's underneath, because it could be cheaters are underneath. But we found that when we use confocal or 2-photon microscopy, we can only penetrate at most 2 layers and not beyond. So this is quite painful. And so Babak and Momeni figured out a way to freeze the cells, freeze the communities, and do cryosection. So this allows us to look at the Z cross-section. And you can see here, right? So you can see here, like the red, green, again, the cooperators are mixed, but the cheaters are excluded, and they cannot grow as tall as this, right? So there's certainly valuable information hidden in this community structure, but it's not readily accessible. So that is one of the challenges. Okay, so now I started with the part that I feel really nervous about, right? So if I were to be asked, right, what is my wish list for microbes, for other microbes other than yeast? This is my wish list. So every microbe is at least as easygoing as yeast. So that's my #1 request. So what does that mean, right? So I want, it can be cultured in at least one defined and transparent medium. Transparent so that I can figure out, right, whether my medium is contaminated or not. And also can do cell, OD, optical density cell counts very quickly, right? And defining mediums so that I know what they need, what makes them sick. Second, I want it to be genetically manipulatable. So in the sense that foreign DNA can be introduced and stably maintained, and it can be expressed, and also genome can be modified. The second, every organism and its environment can be visualized. It does not mean I will actually necessarily monitor every single thing, but I want to have the option to be able to query this if I want to, right? And we are very far from this. And third is that molecular events in the organism can be recorded in vivo. That's so again, I may not want to see everything, but if I want, I should have the option to see it. So my talk, the next part of my talk will be structured in these orders. And if my time's up, I will just, not go through the whole thing. So it's fine, right? So it's up to Orkan to stop me. So first, how to isolate and culture microbes. It's highly non-trivial, right? And then the second one, again, equally non-trivial, how to visualize microbes and their environments in space. And third, how to record events. So I will start with this. So I want to introduce a concept first, a technique first, called multi-TOF mass spectrometry of microbes. How many of you have heard of this? Okay, half. That's good. So if I make any mistakes, correct me, right? So what is that, right? So it's matrix assisted laser desorption, ionization, time of flight. So what happens is this. It's basically it's a big this kind of cylindrical instrument. So what happens at the bottom? Let's start with the bottom. So basically, you embed communities, right, in this matrix. And this matrix will help to extract cellular proteins, such as ribosome. And then you have pulse. You pulse this with UV laser, right? With UV laser, so that basically it slices off these analytes with the matrix and the cellular proteins, and then fragment it and then ionize it. So it's now charged, and then go through this electric field to accelerate. So at the second stage, there's no electric field anymore. So it's drifting of these. So there's a detector at the end. So any ions that have a small mass to charge ratio will fly first. They will reach the detector first, and these other ones will reach later. So then you get a spectrum, right? Called this M/Z spectrum. So then this is matched with a library, with this database, and so then with the known signatures, right? So then we can identify microbes. So the advantage is it's very rapid. It's less than an hour, and it can precisely define the genus and species. So in fact, clinicians use that to rapidly type, right, pathogenic bacteria. And it's actually kind of neat in the sense that they can develop it further to do very rapid antibiotic susceptibility test. So what happens is that you can use this to detect, say, degradation products of antibiotics or enzymes that are capable to degrade antibiotics, and also, growth via isotope amino acids, basically the ability to grow despite the presence of antibiotics. So these are the applications. So caveats, or I don't know, you can, depending on how you look at whether the glass is half full or half empty, it requires 10 to the 4th to 10 to the 5th cells. So it's a very tiny quantity. So in some sense, it's good because you don't even need many cells, but it definitely needs more than one cell for sure. And it cannot distinguish closely related strains. And there's limited database for filamentous fungi. So these are the caveats. So this then has been used to have this culturomics, right? Because everybody wants omics, right? So this is culturomics. So these authors, right? So the authors, they try to diverse, right? So over 200 different culturing conditions for gut microbes, because gut microbes are dominated by some, like say, E. coli, right? So first they will eliminate, right, dominant populations with lytic phage. It's size separation, pre-culturing in aerobic and anaerobic, or anaerobic blood culture conditions, and culture in sterile lumen fluid and sterile human stools with or without this. So they tried all these conditions, right? And then, the last one I thought was really cute. So they co-culture with amoeba. And so that would enrich for these rare microbes that are, the microbes that are low abundance, but are resistant to amoeba predation. So for this, it's actually out of the 200, so 70 conditions, 100% of gut microbes can grow, right? So 20 conditions, 73 can grow. So this is 73% can grow. So these are so culturing conditions can be simplified. So then they did the Molotov mass spec, right? So sort of like colonies of 100 to 300 microns. So because sort of like you cannot see by naked eye, right? You have to use a magnifying glass to see that. And it's high throughput. They can do 32,000 colonies. And then, so at the same time, can do sequencing, right, to identify the based on 16S RNA to like type of the species. So they define the same species as the if they share at least the 98.7% identity. And so they identified 340 species, out of which about half are new. So it's quite remarkable. And if what's surprising is that if you look at the intersect, right, for culturomics for 16S, the overlap is actually quite small, right? So that actually tells us these are still huge unknowns there. Any comments on why there might be such little overlap? Also, I want you to interrupt me, right? Even though I'm not expert, but there are experts in the audience, right? So if you have questions, just interrupt me. So why do we think? Why is there so little overlap?
Kind of unculturable species are detected by 16.
That's right. That's right. But what about the others? The other way around. Why it's so little? Right? Why it's not? So like then I say, well, why is because it's yes, if it's non-culturable, then it would be here. It would be here, not here. But still, then I ask why 16S didn't show up there?
Enrichment.
Why you do regular 16S? It doesn't show up in the culturomics.
Enrichment of very rare taxa that might.
Yeah, that's one part. I also I wonder whether 16S it requires you to be able to extract DNA properly, right? And the culturomics is a different way of extracting. So I wonder, right? So it's just I'm just going for freestyle discussion here. So because I don't like a quiet audience, right? That's very detrimental to our mental health. So yes.
Maybe I missed something in the transition from the first part of the talk to this part of the talk.
Yeah.
You showed us nice experiments in yeast, and then some of the difficulties you have reasoning about those experiments.
Yeah.
And then we switched to pretty different experimental methods.
That's right. It's an overview. That's the introduction.
But in what spirit are we talking about is this about quantifying the composition of much more complex communities than the ones you've studied, or is this about finding new species to work with, or methods?
That's a good point. If we want to measure something, I argue that if you want to measure something, it's beyond the superficial level, you will need to have a lot of materials, and you need to know what it is. So that's why I start with culturing first. So as a half microbiologist, that would be my wish number one. Right, because the reason I can do the yeast experiments is because I can transform them. I can tackle them with different fluorescent colors, and they don't stick one around chemostats, like all these nice features, right? And so I can only imagine for microbes that you have difficulty culturing, then you have to rely on, I don't know, it's very hard to do, experiments that reveals mechanism. You can do correlation experiments, but I think mechanistically it will be very, very hard. So, yes.
So this data is based on this paper from 2012.
Yes.
So I think in the recent years, I
will. So that is my to hold for 1 second. So don't spoil the one joke I planned here. Okay. So genome sequencing of new species. Why they didn't do this? Okay. So that's the paper, right? So then 2016, so these are to me, it's just really amazing. And they can do technical. So they have the Nature microbiology paper, right? similar, a similar flavor, but much larger scale. But now it's my surprise. I'm like, oh my God, retracted, what does that mean? So I looked up. So the authors were not able to provide documentation of approval from appropriate ethics committees, and thus the editors retracted. So basically, I don't know what to think about, but I would say this is article, this is probably the first and perhaps the last retracted article that I feel that you should still read. Right? It's just really striking. But in this experiment, they just did before, but it was much bigger scale. And again, they discovered it's just really amazing. 50% of species are new, right? So there's just immense diversity there. Okay, so then, but about genetic manipulations, right? So these authorities, I mean, these are like the experts, right? They say without a major breakthrough, right, the genetic tractability of microbiome remains arduous. And unfortunately, they're semi-tamed, I call them semi-tamed microbes because they are genetic systems. And I think some of the folks in the audience have worked with at least one or several of these. So we look forward to hearing from them. And the difficulty is to, again, have DNA being able to introduce, being able to be stable, stably maintained, and then be able to express, right? So, and you have to overcome host defense system that try to destroy foreign DNA and so on and so forth. Okay, so the second part, how to visualize microbes and their environments in space. Yes.
If you are finished with the first part, so do you think is going on with this mismatch between culturomics and 16S? Is it just 16S doesn't sequence deep enough? How does it depend on the depth of the 16S sequencing? I thought that 16S captures everything.
Exactly. So who are there? Because I have actually not done any 16S. I'm really ashamed that I have not done that. Right. So so any comments from the audience? Yes.
I think in general, your point about DNA extraction is perfectly valid, but if you do enrichments, as was mentioned with 16S, you see all these things come up from below the limits of detection. I just think, the reality is that communities have a sufficiently complex environment that they can support. We think of like 1 in 1,000 as being low abundance, but we're 1 in 8 billion.
That's true.
I would add to that the problem, it also comes at that level of, like, when you have a rare species at very low abundance, yes, the solution is you would just do more in depth, but there's always gonna be some that are rare enough that you are not safe to know that they're not contamination coming from elsewhere.
That's right.
And all of the process has some low-level contamination.
Actually, that's right.
And so usually you discard it.
Applies to culturomics, right? Because they don't really protect it when they start culture, and it can kind of come out of thin air.
But they say they pre-cultured in sterilized stew and the luminal fluid. So I don't know. Of course, anything can happen, right? I mean, anything can happen, right? So, okay, so how much time do I still have?
If you want to count your 30 minutes, you have 10 minutes left.
I see. Okay. So coming to anaerobes, right? Because a lot of microbes, a significant fraction of them are anaerobic. So anaerobic means those cells don't like oxygen, right? So so the classic fluorescent proteins, they require that we use, right? And I would say CFP in my laboratory is our experience. Our experience says actually it's better than it's less good than BFP. So we have been using BFP. So these fluorescent proteins will require oxygen, right, to fluoresce. But anaerobes cannot grow in the presence of oxygen. So what have people done with these? So like there's techniques, right? So these is first class is flavin mononucleotide binding proteins, but they are very weak, so not so useful. And there are other ones that ligand, right? So ligand not fluoresce, but when they bind, it fluoresces. But then it's not stable in a sense; it's reversible. Right. So for this, you have different color ranges, but it's not very stable. And then there's another category, which is irreversible binding. But the problem is it's also you have very big, spans a lot of spectrum. but the ligand will autofluoresce. So each seems to have its own demo, right? And but regardless, it's still like this is a cute experiment, right? So when you have anaerobe, like it's red labeled and green labeled, and they can fuse, right? Because these two types can fuse. And then if you want to measure the fluorescence, you have to start to add the ligands. And then you subject them to imaging. So this is facts. And you can see, right, the green cluster, the red cluster, and the hybrid cluster. And the microscopy, you can also see this red, green, and the combined. So there's some hope. And then I want to move to, imaging communities, right? Imaging communities. So the classical way, the traditional microscopy goes like this, right? You have light comes in. So let's consider 3 points, A, B, C, right? So A and B on the same focal plane, but C is out of focal plane. So, but then the entire sample is exposed to the light, and then only the A, right? The light from A would go through a pinhole, and the other lights are filtered out, and then it gets to the detector. So this process is actually it's really toxic because C is not to be imaged, but point C is still exposed, and inefficient in the sense that you only see A, you don't see B. So then there is so the light sheet microscopy. Instead, they focus the light and into a sheet, right? So C is no longer exposed to toxic light, and so then you can look at many points simultaneously. And so this is the setup in Raghu's lab, and who unfortunately could not be here because of teaching duties. So because of its orientation, so they have to be orthogonal to each other, right? So that creates some problems with sample mounting. But regardless, right, you have excitation, temperature controlled this, and then collect emission. The emission light is. So the advantage is 3D, and it's a large field of view because you're looking at the sheet at a time. And it's faster acquisition and a long observation time because of low toxicity, because you're not damaging other parts. So they looked at the larvae, larval zebrafish, which is actually transparent, got imaging. So the volume is on a millimeter scale, and the duration hours, which is very long to capture bacterial growth dynamics. And the resolution they want to look at a single bacterium resolution. And the frequency is every 30 seconds. And so in their case, it happens to be the peristaltic contraction frequency of the gut. So this example, but for time purpose, I'm not going to show the example. Yes, Cole.
I guess maybe I'm a little bit repeating my previous question, but so for example, with GFP,
yeah,
it's not like there's zero oxygen in an anaerobic chamber. There's some nice papers showing that you can get GFP to mature in an anaerobic chamber because its maturation is first order in the oxygen concentration, but there's non-trivial amounts of oxygen even in my anaerobic chamber. You can also true? Yes, yeah, it's true.
I see.
It just matures slower.
I see.
But I'm trying to make a larger point here that you can also, there are options with GFP also that you can take it out of the anaerobic chamber and put the cells in the fridge and then
that's true.
And the point I'm trying to make is not that like it's wrong or anything, but just that it seems to me like the experimental methods are contingent on the question, and many of these problems are not exactly problems for certain questions, right? Like if you can make a single time point or staged experiments or whatever. And so I'm sort of curious, like, what are you imagining as the questions?
For each example, so this is not, right, so I was tasked to give introduction, especially for mathematicians. What are the state of the art, right? Like techniques for doing experiments. And I just give some examples. So that's what I'm saying. It's very different from normal talks I would give, where there's very clearly defined questions and I know what I'm talking about. So this is sort of like just like eye candy, like window shopping kind of like, oh, this is cool, that is cool. Sort of like it's like, as I said, it's a random walk into the landscape of this. So for this, you can imagine questions can be answered. If you can look at you can imagine questions. So maybe I will ask your question. Given this, what questions can you address? I will turn the question around.
Yeah, I don't know. I've thought about it, about light sheet.
I can tell you what the author did.
Yeah.
So with the light sheet, they could look at, for example, colonization dynamics of microbes, and then how does it change when the gut contracts, right? So, but without this kind of resolution, they won't be able to track, say, growth and movement and so on and so forth, right? So basically allow this in vivo imaging of gut microbiota in that case.
I guess what I'm saying is I feel like there's sometimes from the experimental, maybe this is part of the theory experiment gap.
Yeah.
Myself mostly doing experiments, is that sometimes we have a kind of maximalism where we're like, oh, we have to measure everything.
That's exactly what I said at the beginning. It's not like I want to measure everything, but I want to have an option to measure if I want, right? It's different. If you have no option to measure something versus you don't want to measure, it's very different. When, it makes me feel helpless. I really want to know that, but I cannot do it. It's just really frustrating. I'm sure you can sympathize because you're experimentalists.
Yeah, but I also find that there's power in giving up on some things to gain other things.
True, and I think Akshay is going to talk about that, right? That's why we have this thing. But again, I think if God asks me, do you want to have the technique to do it? And you can choose to not use it. I would definitely say yes. I will never say no to a methodology that is available. I can always say no, I'm not going to use it. But it's a case, yeah.
Just to sort of add something in between here. I mean, there's an interesting perspective article by Andreas Baumler about how the microbiome field needs more actual microbiologists. And I think one of the things that chases them out of the field is the lack of tools to actually understand what the cells are doing. And, I would say just maybe in to try to answer these questions that, we've gotten so much out of fluorescent microscopy to understand the subcellular dynamics of what powers cell growth. And it's very frustrating not to be able to do that for the whole host of microbes that we care about in the gut. And I think that would actually also have the net effect of drawing more people into the field.
Yes, okay.
Two quick points. One in defense of Wenying's talk. I think the idea was to also just sort of get the discussion starting with these talks. It's experimental, right? So, and then the second point is to what you're saying, which I thought was interesting. You mentioned the sort of maximum. How did you call it? Maximalism. I guess the opposite point could be made as well. Like we tend to go for things that we can measure because we can measure them, but are they the things that we really care about? Like, for example, not to make a blunt point, but like we can study E. coli under rich conditions and highly aerated, but what is the ecology of E. coli? And maybe it is sort of microaerophilic or highly fluctuating and so on. So I guess it is an interesting discussion point. I agree with you that we shouldn't be measuring everything, but also.
Yeah, I know that is a really good, like what's the coarse graining? Because several of us, right, are thinking about coarse graining of ecological phenotypes, right? How do we coarse grain these? And and again, if the coarse grain, I would want to be able to quantify it still, right? Because that's how you test the model predictions. If you cannot measure it, how do you test the model prediction, right? So because of time, I'm not going to show that, right? But the caveats, of course, there's very sample dependent transparency. It won't work for yeast, for example, right? And the 3D reconstruction is not trivial. It may not be able to distinguish individuals if they're in cluster and the host background. So I wonder, like I can stop at any time, right? Because it's just
Finishing 2 minutes.
2 minutes. Okay, so good. So one example is MALDI, again, going to MALDI, like they call it in vivo, sort of like, they call it like a mass, like metabolite and the species, right? Like simultaneous imaging. So it's a deep sea muscle. So this example, the model system, and it has intracellular symbionts. And these intracellular symbionts basically fix carbon dioxide, use energy from methane oxidation, so it's MOX, or sulfur oxidation, SOX. And then the way they do it is, again, they want to understand host microbe interactions, right? So they do this mass spec, sort of like a MALDI, the similar MALDI talk we heard before. And then, but this actually achieve 3 micron resolution, but microbes are only less than 1 micron. So it's still not good enough in the sense that cannot. So the supplement is information by fluorescence in situ hybridization called FISH, right? So fluorescence. So what you do is that you couple these probes against 16S RNA gene with some fluorescence. And the different species can have different fluorescence colors. So then you can look at the spatial distribution of microbes. And then you get images, right? So these 4 panels, like each is a microbe, each is a metabolite, right? The distribution. So the heat map is a percent, like it's like a relative ion intensity. So you can see the patterns are different. And it's a bright field, this is a fish, you can, so it's like the 2 symbiont and the host. And so then you can do the segmentation kind of thing, right? So like the microbe, you know, the metabolites like this, and the symbionts, you can, the red and the yellow, the pink and the yellow are the symbionts, right? And then you can then do like this networking in the sense that, because for metabolites, when you detect the metabolites, it's actually not trivial because a metabolite can have derivatives, right? You can add a group or subtract a group, but they're all related. So that's why they call this MS molecular networking, to identify those and then verify by LC, liquid chromatography, MSMS. So now this is the results section, right? So like they have this, they have this, these are the 2 important cell types is ciliated edge cells. These don't have symbionts, and the bacterial sites, which has symbionts. So what they did is that it's actually really beautiful, right? So you look at this, look at the DNA that stains everything, and you can clearly see this edge cells, the CE, don't have any symbionts, right? But the other bacteria, so BC has a mixture of the two types. And if you look at the metabolites, right, you can see the host-only metabolites because it's present in the CE and absent in the BC. And you also see symbiont microbe-only ones, right? So which is opposite, which is opposite pattern. So then we'll hear more examples from Michael in the next talk. So I will stop here because my time's up. Any more comments? And we have a lot of discussion times, and I would love to hear, right, a discussion on, you know, like how do we choose what to measure, and how do we figure out how to measure them? Yes.
It seems like the models should guide what we need to measure, right? So like usually if there's like
I don't know, like because you know I'm like because many times, right, because there are cases in the history of science, right, it's you see something and then the question arises, and that's why microscope in the initial invention of microscope is so powerful, right? So like you're right, like the model prediction will lead you to measure some things, but I was even seeing things. I mean, I'm trained as a molecular biologist, so there are many cases where you see something that helps you to formulate a hypothesis, right? But it's true, it's quite untargeted if you just go look and see what happens, right? So
I agree with Ophelia. I think the one thing I would say about this maximalist debate is that sometimes the presence of the tools begat their usage, even in the absence of the question demanding an answer to the measurement. So the ability to generate new methodologies to make measurement unquestionably transformative, but in the presence of a question, making said measurement is not always necessary to answer that question.
yeah, absolutely.
So I would prefer not to make the measurement at all costs. And
No, but
if theory friend comes and tells me I have to know that number.
Are you trained as a theorist?
No. I'm trained as an experimentalist, but I'm lazy, so I don't want to have to.
I feel like that's putting a lot of faith in theorists. I of course respect theorists a lot, but I don't want them to, I don't know, because they can often be wrong, right? So I don't know, like, so that's been my career. At the mercy of theorists, and I kind of don't want to do that, you know, like it's personal choice, right? And like, I don't know, like super resolution microscopy, I remember, right, when this first came out, people saw so much more detail, and it fueled an entire field of asking new questions because they see things they normally could not see. And the invention of GFP, right, like it's exactly the same, sort of like going back to Casey's point, right, that allowed us to see so many things we could not otherwise see.
Again, I 100% agree with the development of technology.
Yeah.
What I'm saying is presence of question.
Oh yeah, it's even more. Then your grant get funded, otherwise it won't get funded.
I mean, it feels like it's just a double-edged sword, right? And I didn't mean to like, I don't know, impugn your talk or anything. I just trying to understand. But I feel like all of us are familiar with the way in which the development of sequencing technology has been a double-edged sword for our field, where we have this massive overflowing endeavor, right? Most of which is borderline nonsensical, right? Like just like tons of whatever PCA, TSNE, whatever.
I agree. I completely agree with you.
And it's like self-justifying. It's like, oh, we did the sequencing, therefore we get the paper, and therefore,
Yes,
right? And there's the whole pursuit is in pursuit of nothing, as far as I can tell.
like,
Agree.
well, like, well, I didn't have time, like, tell the authors of those papers, right? So,
No, but like here in this room, I think we can sort of like try to dance on the edge of the sword, right? Like try to figure out where
I
things
agree.
go.
Like, I don't find these papers very informative at all. Like, I completely agree with you. But there are examples I didn't have time to show. There are examples where they claim they could see things. And actually, that was my last slide, right? The last wish is every talk, every paper talk about caveats. That's my request for the rest of the workshop, right? So let's all talk about the caveats. Yes, go on. I mean, is my Q&A over?
You can go on for a few more minutes. Yeah.
Do you have a question?
Big question should be when the model.
Yeah, let's go for it. Because
Yeah.
Akshat has.
Kind of how science kind of works. You find a model doesn't predict something, you kind of hammer away at it. It's like
I cannot hear you. Sorry.
like if you said like what questions do you ask? Well, the questions you ask is when the model's wrong. The model doesn't fit something.
No, it's not this, right? It's like, yeah. Do you have an answer for?
I mean, I see both points, right? Like I see on the one hand that if you have a clear model and a clear prediction that tells you what you need to measure, and then you can even try to figure out a method to measure that thing that you really need to answer the question. On the other hand, I mean, history of science and technology would show that there's a lot of things we couldn't even imagine we could think about until we had the technology to measure them. And so there's a little bit of a tension between these two poles.
Chicken egg problem, right? And another thing is that model, right? You need experimental measurements to constrain the models, right? Otherwise, the model is garbage. I mean, as far as I can tell, it's more or less garbage, right? Because you can put all sorts of assumptions in there. And how do we know, right? These quantities are like even at the right ballpark, right? So that's my, answer to that. For synthetic systems, I completely agree, right? Because we know, like the cosmos, right? We know what's going on, so we can have this. But for like something mysterious like the gut microbiome, I'm not sure how I can think of like modeling. Maybe Casey can, or other bolder folks can explain. Yes.
And I feel like, some of these measurements, if they're not quantitative enough.
That's right. I agree.
Then they're not as useful. And the other thing is, often we need a lot of throughput, right? With the measurements to be able to. Absolutely. So if you're just like one measurement and it's super hard to make, it's not gonna be very useful.
Absolutely. And also scale, right? Meanwhile, just setting up, so I just keep talking.
Yeah.
Right. So the scale, right? So the bridging of the scale, because when you do really these like single cell kind of measurements, you don't have many molecules to measure, so it's very, very noisy, right? But then you have to bridge it to the sample scale, right? The sample is bigger, and then to the whole community scale. Okay.
No, go on. Yeah.
No, I'm done.